now we're going to talk about calcium and regulation of that. So if you want to remember your normal calcium levels, right, calcium in your blood, if you call 911, right, it's between 9-11 milligrams per deciliter. So less than 9, you'll be, on the low end. Above 11, you'll be on the high end. Another way to say that, above 11, hypercalcemia. Below the 9 will be hypocalcemia. What regulates the calcium levels in your blood is the parathyroid gland and the thyroid gland. And the thyroid gland secretes calcitonin, and your parathyroid gland secretes this parathormone. So we're going to go through one by one, explain to you how they work. So here they give you an example, right? So they say low calcium ion levels in the blood, right? So they give you an example, 8.5 milligrams per deciliter. And we said 9 to 11 milligrams per deciliter is your normal range, if you want to remember it. So here we have 8.5. So your body says, okay, I have low calcium level in my blood. I have to find other ways to basically replenish those stores. So what it's going to do, it's going to say on the parathyroid gland, which is the small dots here, and what they secrete is this hormone, right? So the parathyroid hormone is being released. And what does it do? First of all, it's going to come onto the bone, and it's going to work right on the adjacent cells of osteoblasts, and it's going to bind there. And what it's going to do is going to stimulate them, and it's going to then stimulate these cells, which then going to signal the mature osteoclasts to basically break up and release the stored calcium ions. So there's bone resorption, and calcium is going to be released into the bloodstream. So that's one of the effects. The other effect that this hormone does, it works on the intestine, right? And it enhances the reabsorption of calcium in your intestines, right? So parathyroid hormone works on the intestines. Also, there's another hormone that's going to be secreted by the kidneys. We're going to get to it in a little bit, called calcitrol. And it also helps us, right, reabsorb more calcium, and, right, that will elevate the levels. In the kidney, right, the nephron, the moment, right, the parathyroid hormone is released, it's going to act on the kidneys, and the kidneys secrete something known as calcitrol. And this calcitrol, right, helps us, right, to reabsorb more calcium, right, into the interstitial space from the kidneys. Again, calcium reabsorption, so less of it is being removed from the kidneys and the urine, will help us increase the level of calcium in the blood, right? Where do we use calcium normally in our body, right? So if you recall, we used calcium a lot of times. It plays a role in muscle contraction when it binds to the troponin, right? Your muscles are going to contract. Where else calcium is used? We talked about, right, in your, for example, cardiac cells. There was depolarization sensitive SA node, AV node that's using calcium to basically cause the cell to depolarize, right? So we see that in muscle contraction, we see the propagation of the impulse in our heart cells, right? Just some of the examples where we see the calcium plays a role. So this is what happens in our body when we have low calcium levels. Let's see what happens when you have high calcium levels. Let's say you have high calcium levels in the blood. It's above 11. There's a different hormone that's being secreted. It's called, right, calcitonin, which is secreted by the thyroid gland, right? So the thyroid is this big gland here, right? So calcitonin is released. So what happens, right? So calcitonin here, they show you that it increases the resorption of the bone, right? So less bone is being broken up. Less bone being broken up, less calcium is going to be released. In addition, right, if you do not have this parathyroid gland acting, right, there's less of it being reabsorbed in the kidneys and in the intestines. And also, if you have less parathyroid hormone because it's not being released, there's less of this, right, acting on the kidneys. So there's no, I should say, resorption of calcium. So increased calcitonin level, which is secreted by the thyroid gland, right, it inhibits the kidneys from reabsorbing calcium into the interstitial space. So more of it is being lost in the urine. So all of this combined, right? Less bone breakup, less calcium absorbed in the intestinal tract, less of it being absorbed by the kidneys, and more of it being excreted in the urine, right? Maybe we can, right, fix our high calcium levels in the blood. So the takeaways, right? Remember the two systems at play, right? The thyroid secretes the calcitonin. When is the calcitonin secreted? When you have high calcium levels in the blood. The other one is the parathyroid. That's the small glands on the thyroid. They secrete the parathyroid hormone. When is that being released? When you have low calcium levels in the blood. So hopefully more of it is going to be, like more bone is going to be broken up, more of it is going to be absorbed in the blood. So this is our different responses. And before we continue on, any questions here? All right. So the next thing we're going to get to is the muscle, particularly, right? How is that calcium is used there, right? And talk about all that. So we have our division of the central nervous system, which is the brain and the spinal cord we see here, right? And then if we subdivide further, right, we have the peripheral nervous system. We have the sensory, which is the afferent system, and we have the motor, which is the efferent system. And the motor system, the efferent system, subdivides into two different types. We have the somatic and we have the autonomic. Right now, we're going to focus on the somatic system, right? That's the voluntary muscular control. And then the autonomic system, as you guys are aware, has the two division, as the sympathetic and the parasympathetic division, right? The sympathetic and the parasympathetic. We'll talk about more of that when we get to the neuro chapter. Now we're going to focus on the somatic system with the muscular skeletal control. So you see it here, right? And let's talk about the neurotransmitters that are being released. So we said the somatic nervous system is under our voluntary control, I should say. And that's the system that, let's say we say, all right, contract the muscle and, I squeeze my arm, right? What's the neurotransmitter that's released? It's the acetylcholine that's being released. What's the receptors on the musculoskeletal sites? That's the nicotinic receptors. So acetylcholine, right, and is going to be working on the nicotinic receptors. And you see, right, the end neurotransmitter is acetylcholine, is going to bind here. The other ones I'll just touch on briefly since I have it here, but I'll get more in depth of it once we get to the neuro chapter. So the autonomic nervous system, which I said is composed of sympathetic and the parasympathetic division. The preganglionic, right? Preganglionic is usually acetylcholine, so usually that's what they are. And the postganglionic, right, after this ganglion, is usually norepinephrine for the sympathetic division. You may recall, right, fight or flight system. This is the sympathetic division. And what does it release? Usually we tell you, right, adrenaline, or, we could say norepinephrine. And here they show you, right, the adrenal medulla, which releases both epinephrine and norepinephrine, right? And the response, as you guys recall, right, let's say if it's going to act on yours, well, I should say here, it's going to go into your bloodstream, which is going to be epinephrine. And then when it acts on your smooth muscle, right, we see that vasoconstriction response, right? Alpha-1 receptors, right, and vasoconstriction. That's what we see. The parasympathetic division, right, we see both that the neurotransmitters here being acetylcholine, right, that it's being released. And here they're showing you the heart. There is muscarinic receptors on the heart, M receptors, right, M1, 2, 3 receptors. And what they do is they are the opposite of the sympathetic division. The parasympathetic system, right, they tell you rest and digest. So if you have acetylcholine, right, working on the heart, on the M receptors, it's going to slow down the heart. So you're going to see, right, like bradycardia start to develop if you have like vagal response, right? They say vagal response. So it'll slow down the heart rate. So the takeaways here, right? The somatic system, the neurotransmitter being acetylcholine, the receptors are nicotinic in your skeletal muscle. This is our voluntary system. The autonomic nervous system has two divisions, the sympathetic and the parasympathetic division. This is not under our voluntary control. The sympathetic system is the fight or flight. the major neurotransmitters there being norepi and epi. And the final response, right, depending on the site it basically works on, alpha 1, vasoconstriction, beta 1, increased heart rate, right, beta 2 on the lungs, bronchodilation. And then we have the parasympathetic division with acetylcholine. And the response there is basically rest and digest. So if you have acetylcholine acting on the cardiac muscle, M receptors, right, it's going to slow down the heart rate.
All right. So basically what you're saying is that for the sympathetic nervous system, norepi and epi is both the transmitters.
Yep.
But for the parasympathetic nervous system, it's only norepi.
That is correct. That is correct, James. Parasympathetic, you're saying acetylcholine. Parasympathetic is acetylcholine, James. I hope.
Isn't that? Yeah. All right.
Let me just restate so everyone's clear. For the sympathetic division, right, the post-ganglionic, the one that's actually working on the site of the receptor that we are focusing on, like alpha 1, beta 1, and beta 2, is either norepinephrine or epinephrine. This is the sympathetic side, right? So the only ones that are actually exerting the effects that we are interested in is norepi and epi. For the parasympathetic side, the major hormone that plays acetylcholine. And acetylcholine, right, depending on the site, it targets, like, for example, here being the heart, right, M receptors on the heart. When it binds, it's going to slow down the heart. So the major neurotransmitter of the parasympathetic division is acetylcholine. Hopefully that's clear.
Yeah, I got it now.
Okay, thank you. And here, right, just to show you some of the effects, right, we talked about, right, for example, acetylcholine. Let's talk about muscle, right? We said in here, right, somatic nervous system was under voluntary control. We have acetylcholine is the neurotransmitter. It's released. Nicotinic receptors on the skeletal muscle. So here they show you a neuron, right? And we said we see like the actual action potential come in, and we see calcium is coming in, right? So the role of calcium here is what it does. It causes exocytosis of this neurotransmitter. So as calcium comes in here, right, it's going to trigger this to bind to the synaptic cleft, right? This vesicle is going to bind and it's going to release its contents. So here we see this vesicle coming to the surface. It's bound to the membrane, and then it's releasing acetylcholine on these nicotinic receptors or N receptors at the synaptic cleft. And then this is going to exert the effects downstream, which we're going to talk about in a moment. But basically, this is going to cause depolarization to occur. So what we basically see is calcium's another role, right? We talked about normal range 9 to 11. We also see calcium plays a role in neurotransmitter release, or I should say exocytosis of this neurotransmitter, which is bound in the vesicle. And the moment this neurotransmitter is released, it binds to the receptors, right? And it's going to cause, right, the downstream effects, which we're going to talk about in a bit, right? So essentially what it does is it's going to cause depolarization of these cells. And if you guys recall, right, how does this work, right? As this occurs, we see, right, this lovely diagram, right? This is particular to the muscle. We have one for cardiac, we have one for neuro. But in a nutshell, what essentially happens, this is all mediated through ion channels, right? So sodium-gated channels, potassium-gated channels, calcium-gated channels. And in a nutshell, I think I explained this like several times. But let's see if you guys, I mean, I'll try to do it again. If you have any particular questions, we can go through it. So in this particular cell, they show you resting potential at 70 millivolts. So it means the cell is resting at this range. At this portion at negative 55 millivolts, specific voltage-gated channels will activate. And the moment they activate, it's like all or none response. It's not like one activated and, you know, maybe something else will be activated. No, the moment you reach threshold, it's like everybody's ready to roll, right? It's all the channels are now open. So here we see, right, the arrow shows you voltage-gated sodium channels open, positive sodium ions rush in. So essentially, if you recall, right, we have these voltage-gated sodium channels. I'm going to show you the configuration of them. Usually when they're in this resting state, they look like this. You have the activation gate that's closed and an activation gate that's open. We have some leaky channels that allow trickling in of some cation. So maybe some sodium or calcium trickles in, and as they're trickling in, they're going to raise the resting state to the threshold. The moment this portion is reached, this sodium gated channel, voltage gated, is going to switch its configuration. It's going to look like this now. The activation gate will open and this will start to shut. This is the inactivation gate. So sodium, right, can start to come in from the outside to the inside. And we said cells are bags of potassium and a sea of sodium, right? So as the sodium is rushing in, we see how it takes this from -70 to -55 all the way to +30. So this we call depolarization. So which ion was responsible? It was sodium channel in this particular case, right? So sodium channel and sodium cation was responsible for depolarization. Now you reach here, you notice it says, okay, so voltage gated sodium channels start to close, right? And voltage gated channels open. So what we basically see the moment we reach here, right? This gate, right, starts to close, and while this gate is still not fully shut, right? But we can see that sodium cannot now come in, right? So these channels starts to basically make this a little bit clearer. So sodium channels cannot rush in, but at the same time we have the potassium channels. Let me take a different color. The potassium channels open up, and the potassium is rushing outside because we said cells are bags of potassium and a sea of sodium. So what we see now as the cell is losing positive cations in the form of potassium, this starts to repolarize, right? All the way down here. And once you reach this threshold, you're back to the resting state. Let me take a blue color again. And then you're back to this form, right? From this, so then you're sodium channels take this form again, so they're ready for the next impulse to come in. And again, this signal propagates from cell to cell, right? And we see this depolarization followed by repolarization occurring, right? So if the question is asking you what ion is responsible for depolarization, usually it's the sodium channel, unless you're dealing with the SI node or AV node, which is calcium channel. If you're talking about repolarization, that's usually potassium sensitive. So potassium is responsible for repolarization. So depolarization is sodium, repolarization is potassium. All right, so these are the channels. This is basically how this impulse propagates, the depolarization stimulus, right? How it moves across, right, the action potentials. And now, right, when we have this depolarization, all this is occurring, we also see, right, this actual muscle contraction we talked about. So let me go through this with you step by step so you guys can understand. So we have the electrical followed by mechanical, right? Now, I think you guys, since you had the cardiology, kind of now understand, right? We have the electrical coming in of these ions, right, causing depolarization, and then we have the actual mechanical contraction of the heart. It's kind of very similar in the muscle, right? But slightly different names, right, that are involved here. So let me show you what we talk about. So the way this process starts, we have a thick and thin filament. So myosin being thick filament, this being the thin filament. How to remember? You see the word myosin, right? It's kind of more letters than actin, so maybe they'll help you to remember this is the thick filament and actin is the thin filament. And so before we can activate the system, we have to basically have this myosin head to undock from the actin and go back into the resting status. And the way we do this, we need to utilize energy. Here we use the ATP. So ATP binds here on the myosin, right? And then it changes its configuration back to the resting state here, right? And now we're ready to basically start the muscle contraction process, Other important points I want to point your attention to, right? We see the thin filament. It has the actin, right, where the myosin head binds. It has the troponin, where calcium will bind. And we have the tropomyosin, which surrounded, blocking the actin, right, heads, the active sites, right? So we're going to talk about how this process activates this and, you know, how this whole thing occurs. So let's go one by one. So now we said ATP has bound and it's now in the resting state. So then the calcium comes in, right? That's being stored in the sarcoplasmic reticulum, right? Where does the calcium bind? It binds to the troponin. What is the troponin? Troponin was drawn here, right? So troponin is drawn here. Calcium binds here to the troponin. And this will signal the thin filament to move these, right, blocking sites by tropomyosin away so that the actin heads are exposed. So calcium bound to the troponin, right, signals the tropomyosin to move out of the way. Actin is exposed. And what happens then is, this just shows you the location for this, right? We have the calcium stored in the sarcoplasmic reticulum, right? And as this impulse action potential propagates, the neurotransmitter comes in, right? Sends it through depolarization. This depolarization signal sends, right, the signal to the sarcoplasmic reticulum to start to move calcium, right, from the stores into the circulation here, right? And then, right, as the action potential travels, more calcium is released. This is basically all it shows you. So calcium is released from the sarcoplasmic reticulum. And we see the channels here, right? And then what happens, right? So with calcium, right, what happens? These sites are going to be without calcium, so right now we have no calcium. The tropomyosin, right, is not moved out of the way, right? It's blocking the actin sites. The moment calcium binds to the troponin, the tropomyosin is going to be moved out of the way. So here they show you with calcium comes in, and you see how this actin site is exposed. So now with calcium, myosin can bind here, right? So now the myosin, the thick filament, binds to the actin, and we can have that power stroke. And when it does it, it gets rid of it phosphate group, right? ADP plus phosphate, get rid of this energy molecule. And now we have myosin head that's now bound to the actin. And we see the active power stroke. It basically moves, right, this whole muscle filament. So here we see thin filaments, right, which was the actin and all the other components. We see the thick filaments, which was the myosin. Here we see how it bound, the cross-bridging that's occurring with the myosin head with binding to actin. And then the power stroke, right, it basically contracts, right, and it moves, and the muscles contract. Basically the sliding filament theory in a nutshell. So to combine all of these things together, we got to get myosin, which is the thick filament, back to the resting state, right, or to like that it's ready to contract. The way it does it, ATP comes in, it binds to it, moves it into a resting state, while ADP and phosphate are bound to it. So ATP, when it breaks down in adenosine triphosphate, this is what T stands for, tri, goes into diphosphate plus the phosphate, which is attached to it. The next thing we are waiting for is calcium, which is released by sarcoplasmic reticulum. How does it do it? Through the depolarization, right? That's propagated by the acetylcholine neurotransmitter acting on the nicotinic receptors. Calcium comes out. It's going to look for the troponin, right? When it binds to the troponin in the thin filament, this is being the thin filament, this will then signal the tropomyosin, which are blocking the actin heads to move out of the way. The moment it moves out of the way, myosin head binds to the actin, and you have this power stroke occurring, right? Muscle contracts. Right? Any questions about this? Right. So hopefully that was clear. Since nobody asked me, you guys really clear about this? No questions.
Yeah, all good.
Jacob, you have a question?
No, I was saying that I guess we're all good.
I cannot hear you at all. If you want
Now
to,
he's saying that we're all good. We're good.
oh, okay, all right. I'm just making sure, right? So, all right, if you're clear, that's good. So, the next thing I want to talk about, right, is we talked about a little bit about aerobic and anaerobic respirations. You don't need to remember all the stuff. What I'm going to basically get you to understand is that in a setting of oxygen, we perform aerobic respiration, and usually we make about 32 ATP, right? We see here, right, the net response per glucose molecule, I should say, right? When we utilize glucose molecule in the setting of oxygen, we make 38 ATPs, which we employ, right? As you saw here, for example, ATPs used for muscle contraction. We also learned about ATPs using for sodium potassium ATPase pumps, right? That basically use that energy to move the ions across their concentration gradient, or I should say against their concentration gradients, right? Now, when you don't have oxygen and you switch to anaerobic respiration, right? What you see is that per glucose molecule in under anaerobic respirations, you only make 2 ATP, right? So obviously 2 versus 38 is a huge difference, right? So that's why patients who have anaerobic conditions, not only their cells have higher metabolic demands, right? The cells are not making ATP to fuel other processes like muscle contraction, like ATP, sodium potassium ATPases that we have, and other processes that require ATP. So our job is basically to correct their condition, or basically stabilize their condition, so that it doesn't progress, right, forward. That's why we tell you, right, in a state of shock, right? Some of the basic treatments that we render, we want to maintain their blood pressure, so we give them some IV fluids, make sure they want to keep them warm. We give them oxygen, right, if their saturation is low. So hopefully support them as we're transferring them to the hospital. So takeaway, right? With oxygen, you are in aerobic conditions, right? So let me take a different color here. You make about 38 ATP, which drives all your metabolic right needs that require ATP. No oxygen present, you're in anaerobic condition, right? You can only do glycolysis, right? And because of that, you only make 2 ATP. There's a cell in your body that can only perform glycolysis. It cannot, it doesn't have mitochondria in it to continue with the aerobic conditions. Anyone knows of these cells that only perform glycolysis?
Liver cells.
Which ones?
Liver cells.
Not liver cells. Liver cells actually require, right? And they have mitochondria, and they produce 38 ATP. Not liver cells.
Brain cells. Sorry, brain cells.
Oh, brain is the most important cell, Helmi. Brain cells will definitely we need that.
Cardiac cells?
Red blood cells.
Red blood cells, very good. So red blood cells do not have mitochondria, right? And they only support their metabolic needs via glycolysis. So they can never, do aerobic respirations. They don't have the mitochondria. It kind of makes sense, right? It's like the cell itself, their job is to carry oxygen to the tissues. Imagine if it was using that oxygen, to do its work. It's like the equivalent of that would be like UPS is bringing the packages to your house, right, for delivery. Instead, the UPS was taking all the packages for themselves, right? And so, the carrier molecule should not be consuming what it's carrying. So oxygen, you don't want the red blood cells to consume the oxygen. So the glycolysis is sufficient for its metabolic needs, right? So hopefully that makes sense. All right, any questions about this? All right, so we talked about, right, the somatic system, the skeletal muscle system right now, right? This is the system we spoke about. We talked about the muscle contraction. But now let's talk about, I think, the autonomic system. Let's talk about the two divisions here, the sympathetic and the parasympathetic system. And we're actually going to talk about also this propagation of neurons, right? One thing I will say, if for your medications, specifically pressors, right? Pressors. You really want to understand the response of the sympathetic nervous system on the normal receptors, what it does. Then you could understand how your pressors work. When somebody asked me about the pressor chart, but they usually indicate, right, which pressors affect alpha receptors, right, at what doses and what's the response, right? So when we are looking at what pressor to administer, you want to look at the dose, right? Because, for example, dopamine at the doses of 2 to 5 is not going to give you systemic vascular resistance. It's not going to cause that alpha-1 response, right? You got to get to the 10 and higher micrograms per kilogram per minute dose range before you could exert those effects. So you need to understand that, right, that the alpha response is not there at the low doses. Then you may have other medications like norepi that gives you significant alpha-1 response when you give it. So you need to pay attention to the receptors and at the doses those drugs exert those effects. Right here, it was basically a slide from the different cations, right, that we see, right? And we talked about cells being bags of potassium and a sea of sodium. We see the equilibrium potentials where this wants to take it, right? And I think I drew out the diagram I explained in the one earlier. So this is similar, right? And in the way it works is similar in the cells. However, some cells may use different cations for depolarization. For example, SA node and AV node is not sodium dependent, right? It's calcium dependent for depolarization. Now, Purkinje cells, right, in the ventricles, they're sodium dependent, right? Your neurons, sodium dependent. So depending on the cell, right, the depolarization mechanism may slightly differ. So that's the major thing here, right? And here, right, there was basically the same response I was showing you. This here we're getting into the neuron and the depolarization of the neuron, right? So the resting state, the threshold state, depolarization and repolarization. I think we went through this already. Any questions about this? All right. The other thing that I want to also point out in terms of the sympathetic system, the major divisions when it extends, it goes through the thoracic vertebra, right? So you see it starts with T1 and then, right, basically ends here, right, at L1, lumbar region. And the predominant output of the sympathetic system, right, we see it's originating from the spinal cord, right, specifically at the thoracic vertebra. That's why when you guys assess, right, for neurogenic shock, for example, you notice, right, they will be in shock, but they don't have that rebound tachycardia to go along with it. And it makes sense, right, if you have injury to the spinal cord somewhere here, maybe it cut off, right, the responses, or somewhere here, cut off the responses for this tachycardia on the heart. So you may not see that rebound tachycardia occurring. On the other hand, right, on the parasympathetic side, it originates, right, from here. We see from the pons region, and we see these nerves, right, specifically the vagus nerve, the glossopharyngeal nerve that works to slow down, right, the rest and digest system, right, and we see some of that from the sacral region, right. So the hormones, the neurotransmitter, I should say, that's secreted from this system. We talked about the acetylcholine from the parasympathetic side, and we said for the postganglionic nerve on the sympathetic division was the norepi and epi, right? And this was our fight or flight system. This was our rest and digest system. The reason why I'm showing you this is just to show you where these nerves originate from, sympathetic versus the parasympathetic side. Right? So you see there's no innervation that's coming from the spinal cord to the parasympathetic division. In your body, both of these systems work together. It's not like sympathetic is not working and sympathetic is working. They're all working together. However, one predominates depending on what you're doing, right? So let's say if I'm sitting on the chair, I'm relaxing, right? Both of them are working, right? Because still, you got to have some sort of a vagal tone, right? I should say some sort of a vasoconstriction there, so that your blood pressure is maintained, so the blood flow goes to the heart, to the brain, so you're able to listen. Maybe you ate something, you're digesting that food or something you ate earlier, right? You have peristalsis in your GI tract, right? You're making urine and so forth. And the moment, let's say, I decide to stand up and go to the kitchen and make something to drink, for example, the moment I stand up, the body is going to say, okay, I have to vasoconstrict because I don't want to syncope, right? I don't want to pass out. So there's some form of vasoconstriction that's going to occur, raise my blood pressure so I can walk without any problems, right? That's why those patients who get prescribed alpha-1 blockers, right, on the, for the high blood pressure, like Prazosin, Trazocin medication, they block alpha-1. They tell them, don't ambulate quickly, meaning don't get up out of your bed, when you stand up, because you stand up, you don't have that rebound vasoconstriction, and they will syncope. So that's why it's important to know, right, these systems, how they work. They usually work together. Right, so here we see the actual neurotransmitters, as I was saying, right? For the parasympathetic side, we just explained, right, was the acetylcholine, right? The preganglionic, and also acetylcholine being postganglionic. Here they show you M receptors. And for the sympathetic side, the ones we are concerned is the postganglionic, usually being epi and norepi, right? And this is here that we see that, right? Somatic system, we just talked about, this was the muscle system under voluntary control, and this was the acetylcholine, right, acting on the nicotinic receptors. All right, and the effects, right, so we have all these different receptors. The ones you are responsible for, right, as I already explained to you, right, would be alpha-1, which causes smooth muscle constriction. Talked about beta-1, talked about increasing the heart rate, talked about beta-2, it's a vasodilation or bronchodilation. We talked about in the heart, right, the parasympathetic system acts here, right? M2 receptors is going to cause decrease in the heart rate. So that's pretty much what we talked about. We get to the renal chapter, I can elaborate there, but we basically said, right, the vasopressin, if you guys recall, right, it has receptors of V1, V2, right, vasopressin receptors. And what does it do, right? Vasopressin is another name for ADH, antidiuretic hormone. One goal is, right, to increase smooth muscle contraction, so you basically vasoconstrict. The other goal was to retain water, right, from the kidneys so that your blood pressure increases. So the vasopressin exerts its effects in the V1, V2 receptors. The other questions I was asking, like if you look in your protocols, you do have vasopressin, right? And you have other pressors, right? But sometimes you may see a patient who is on multi-pressors. They may be like on epi and vasopressin, or they may be on dopamine, norepi, vasopressin. So my question is, why have these multi-pressors for the patient?
They work on different receptors.
Exactly, right? So as Aaron says, they work on different receptors. So for example, let's say I maxed out my dose of norepi, right? And I want to introduce another pressor. If I give them epi, right, you're still going to be working on those receptors. I mean, you may not get that vasoconstriction that you're seeking, but I may go to a different drug like vasopressin that works on different receptors. Different receptors are going to give me, right, different response. So that's what they're trying to do with these multipressors, right? To see if they could target other receptors to give you the same effect, basically. Right, so I kind of circle, well, these you definitely want to know. Right, so the ones I circled here is the receptors you want to be familiar with. We went over this already, right? So the next thing we're going to look at is the neuron, right, in the nervous system. And particularly one I want to focus on, right, is these Schwann cells, right, that surround the neuron. And in between these Schwann cells, we have these spaces here, right? So these Schwann cells, they insulate the neuron. So when the nerve impulse propagates, it goes like this through these nodes. And these nodes are called nodes of Ranvier. And the purpose of this is basically you have this what's called the saltatory conduction. This allows to conduct impulses much faster, right? As opposed to going like cell to cell, right? So so this is what we see, right? So that's the conduction. Schwann cells, right? They surround the axon, and they basically like an insulation. They like form these layers around it, right? And they made up of myelin, right? We call it myelin sheath, right? So they surround the neurons. This is isolates the neurons. So the only place where the impulse can be propagated is at these nodes of Ranvier to do the saltatory conduction. And this is what we basically see, right, on this neuron, right? And then we talked about some of demyelinating diseases. We talked about diseases, right, multiple sclerosis. And here what happens is basically it's an autoimmune disease. So body is attacking its own self, attacking its own cells. And this disease process, right, the multiple sclerosis attacks these oligodendrocytes, right, which is not the peripheral, but the central nervous system, right? So oligodendrocytes is myelin sheath that's surrounding the neuron and the central nervous system, and you basically your body attacks these, right? Thinking because it's foreign, right? Something is going on that is these cells are foreign. That's making these antibodies that's attacking the sheath. So you could see, right? If I'm removing this sheath, the impulses are not going to be conducted as readily, not as fast, right? There'll be ataxias and neurological deficits that the patient may have, right? And then also we talked about Guillain-Barré disease. This is basically a similar disease to MS, right? The difference is it's targeting the peripheral nervous system. So not the central nervous system, but the peripheral nervous system, right? And what it's going to do is, again, we're going to make these antibodies that's going to attack the peripheral nervous system and demyelinate them. Demyelinating them, again, impulse conduction is going to be impacted, right? So this is what we see, right? And here to show you some of the symptoms, right? So you may have, for multiple sclerosis, you may have cognitive impairments, you may have, right, poor conduction, right? So this will have weakness, spasms, ataxia, right? It may be changes in your gait, right? And all other conditions that you may see, right, with, I should say, not improper, but impulse conduction that's not conducted through the saltatory conduction, as we saw, right, with those myelin sheath that we had in place. So everything that's involved with not proper propagation of these impulses, right? Any questions about this? All right. So then we will continue on, then we're up to our GI system, digestive system. So the major takeaways here, right, that you want to remember is the enzymes that are secreted and what they do, right? So the major organs that we want to focus here, right, is the salivary glands. They secrete amylase, and that basically chops up sugar. And what's the goal of this, right? So the goal of this is I want to take like a complicated sugar molecule, and I want to chop it down to the individual monosaccharides. Because what I ultimately want is I want that glucose, right? Why do I want that glucose? I want the glucose to drive my anaerobic metabolism, so that this glucose can be turned into 38 ATP, right? How do I, get this glucose? My body produces these different enzymes that chop up these complicated sugars or polysaccharides into monosaccharides, right? And the one I really want, I want that glucose. So we see, right, the salivary glands make the salivary amylase, and then we have the pancreas, right, that also breaks down with pancreatic amylase, breaks down these sugars, or I should say polysaccharides, into monosaccharides. So the goal of all this is to chop up macromolecules into a smaller size molecules that the body can employ, right? We also see stomach makes pepsin, and then this is used, right, to break down peptides. We see the pancreas also makes this trypsin, right? Elastase and chymotrypsin, again, working on your proteins to make them into smaller size, right, amino acids that we're going to employ, right? And the whole function of all this is, again, as I said, take polypeptides or polysaccharides and turn them into either amino acids or turn them into glucose, right? Single chain, right? That the body can employ. The next thing in the GI chapter, what was important, we talked about these different transporters, glucose type 1, glucose type 2, glucose type 3, 4, right? The major ones you want to remember is glucose type 4 transporter. Why was this important? And it's found in adipose tissue and your muscle. Why is glucose type 4 transporter important for you to know?
For treatment, for insulin transportation.
Insulin, right? So when you have insulin that's released by the pancreas, right, it works specifically on glucose type 4 transporters. And when it binds, only then it allows, right, the glucose to then come inside the cell. Up until that point, right, glucose cannot enter the cell. So the entry of glucose to your muscle and your adipose tissue is insulin dependent. However, you notice, right, for the brain, right, we have glucose type 1 and glucose type 3 transporters. They're not insulin dependent. And why do you think that may be? Why is brain, or I should say the central nervous system, not under the mercy of insulin?
Because the nervous system is needed in glucose all the time. If you are under the mercy of the glucose, you're not gonna find the glucose in the brain tissue, like have limited time to be destroyed, like from 4 to 4 and then die.
Yeah.
That's why it needs glucose in continuous, like in continuous way, you know.
Yeah, exactly. So you're correct, Helmi, right? The brain utilizes glucose consistently. And so that's why, I don't know if any of you tried like Atkins diet or keto diets, right? Where you don't eat sugar and you're like in the haze, right? You're like walking around like a zombie. And the reason why is that your brain needs glucose in order to carry out the actions of the neurons and the metabolic functions. That's why it's never under the mercy of insulin, right? So your brain is always in need of glucose. And when it cannot get it, it starts to break up other tissues, right? Like muscle and fat, break them down to get, right, to make glucose from non-glucose sources. We call it gluconeogenesis, right? And that's usually the problem in a diabetic patient, right? Because either in a diabetic patient, either they do not make insulin, they have auto-destruction of the beta cells of the islands of the Langerhans, right? Or they have type 2 diabetes, right? Diabetics where the receptors are not as functional for the insulin. So what happens is body's trying, even though there's a lot of sugar in the body cannot sense it. So it starts to break up, right, other non-carbohydrate sources for it. So the takeaways here is, right, GLUT4 transporters is under insulin, and we need insulin for the glucose to come in. Brain is usually never under the mercy of insulin, right? It always can utilize glucose. They can come in without insulin being present.